Good evening and welcome. It's my great pleasure to welcome you to this AMSI Bioinformatics Summer 2022 public lecture. And I want to start with an acknowledgement of country on which we live and meet here tonight. The University of Melbourne acknowledges the traditional owners of the unceded land on which we work, learn and live, the Wurundjeri Woiwurrung and Bunurong peoples. The university also acknowledges and is grateful to the traditional owners, elders, and knowledge holders of all indigenous nations and clans who have been instrumental in our reconciliation journey. We recognize the unique place held by Aboriginal and Torres Strait Islander people as the original owners and custodians on the lands and waterways across the Australian continent, with histories of continuous connection dating back more than 60,000 years. We also acknowledge the enduring cultural practices of caring for country. We pay respect to elders past, present and future, and acknowledge the importance of Indigenous knowledge in the academy. As a community of researchers, teachers, professional staff and students, we are privileged to work and learn every day with Indigenous colleagues and partners. And I want to follow this with a welcome of all of you to this event tonight. This was made possible by the sponsorship of Melbourne Integrative Genomics, the Australian Mathematical Science Institute, AMSI, Australian Biocommons, and Ginkgo Bioworks. And we're really grateful for the financial, moral, and institutional support that these institutions have offered us. This lecture is being recorded and will be available on the AMSI YouTube channel in the next 2 weeks. If you're online, you can join the conversation on Twitter using BioInfoSummer and tagging @DiscoveryAMC in your social media channels. And you will be able to ask questions at the end of the lecture. And for our virtual participants, you can type your questions into the Q&A at the bottom of your screen. And now finally, it's a great pleasure to introduce Drew Barry. Drew Barry has an unusually rich portfolio of honors and accolades that he has won. I've introduced people with all sorts of things, but never an Emmy Award winner, never a BAFTA Award winner, and never somebody who had collaborated and was praised by Bjork. So for a middle-aged academic, that's about as high as it goes. So Nobel Prize, there you go, but that's much, much higher. In his professional work, so Drew started as a biologist. He wanted to investigate sharks, he told me, but then was entranced by the complexity and beauty of cellular systems. And he's been using his scientific knowledge and his artistic skills to really draw out the finesse, the intricacies, complexities, and wonder of life at the cellular and molecular level. And he will share this with us tonight. I'm really grateful for this because it's a beautiful marriage of art and science that we can enjoy tonight. And we will learn something from the very, very young to the very, very old. And I'm really looking forward to sharing with all of you in this today. He sits very close to us, just across Royal Parade at WEHI, that has enabled much of the work. And we are excited and delighted that Drew will share this work with us tonight. So Drew, thank you very much for coming, and the floor is all yours.
Great. Thank you. It's such a pleasure. And thank you to the organizers for creating the opportunity for me to be able to present to you. We've got quite an array of different kinds of audience here, from kids to older kids. And so hopefully you have the next generation of PhDs coming through here. And I will be pitching to them so that you guys can understand too. So hopefully this will go away, but it's not. Nope. Yes, thanks. We'll give it a go. I'll keep going. Thank you. Will you ever check that out? So I'm actually a creature of this area, this place right here. I have spent the last 30 years probably within about 500 meters of here. I I'm working at the Walter and Eliza Hall Institute of Medical Research, which is just the other side of Melbourne University, behind the Royal Melbourne Hospital. And I've been there since 1995 as their biomedical animator. I'm going to be a little bit indulgent and talk a little bit about my background and how I got into this work. But then I'm going to present some early work, which really was pivotal for opening up my career from 20 years ago, and then some of my more recent work. Mouse pointer. No. Okay, we'll give it a go. So before I was at WEHI, I went to Melbourne University here with the intent to become a marine scientist. And as part of that marine science program, it was compulsory to do cell biology. And I was completely enthralled by this footage here by Professor Jeremy Pickett-Heaps. And I ended up in his lab doing several different undergraduate and postgraduate research into these organisms. So this is the majority of eukaryotic life on Earth, is these sorts of organisms. These are all algae or single-celled organisms. This is a euglena, very, very common all around the world. So it's a single-celled algae, and it has a little propeller at the front, the flagella, which is pulling it through the water. This is another species of euglena. But if you note the little red dot at the end of it, so this is a single-celled organism. That little red dot is a pigment granule that it's a very primitive eye that the cell is able to detect the direction of light to be able to swim towards or away from light. This is cyanura. It's absolutely liquid gold under the microscope. And what you're seeing here is actually from a videotape recording. So really losing all the magnificence of it, but absolutely beautiful. These are multi-celled algae. 4-celled organisms, 16-celled organisms, and so on. So this is where I learned to love cells and spent many years filming cells under time-lapse microscopy, triangular species. There's nothing nature hasn't had a go. And again, these are extremely common. Everywhere you see, after a rain in the slimy gutter, if you put it under the microscope, this is what you'll see. So this is actually diatoms. They have silica glass shells, and these are stacking together and locking together. And this is in real time how they crawl through damp soil. Again, these are an algae, but together with their colonial sliding together, this is how they navigate the world. So how could you not be inspired by these sorts of things? So I was very privileged and lucky to spend my years with Jeremy Pickett-Heaps. This is Volvox. This is a super popular one with an eye of a needle to give a sense of scale. So all of these things are just at the limits of being able to resolve them with your naked eye, but then they become alive under a microscope. So I started in cell biology, but at the same time, I was of the first generation that grew up with a computer. So in my late teens, I had access to various computers, Commodore 64, Atari, but this is really the pivotal machine that changed my life, called the Amiga, the Commodore Amiga. 512 megabytes of RAM. How could you ever possibly fill that up? It was way ahead of its time for graphics and particularly games, as you're about to see. And I was really, really inspired by the video games. Wasted all my summers playing them. Xenon 2, It's a Mega Blast, had an incredible soundtrack, but I was just really blown away by these little organic life forms. How are they doing that? How are they creating these graphics? So Xenon 2, and then when Shadow of the Beast came out, I vividly remember seeing this game and going, it's so 3D. Like, it just can't get any better. They've done it all. It's solved. This computer graphics are done. Like, this is it. And I spent a lot of time cracking these games open, learning the tools and techniques that they were using to build these sorts of games. One of the things I'm going to reference later, how these video games are made with loops, little loops of animation, little loops of behavior, and that enables endless play. So these sorts of situations can go on endlessly. And that is very much a fundamental technology that we've employed in our recent animations or recent productions. So this is me in 1998 when I founded WEHI TV at WEHI. There's a zip drive that was 128 megabytes. I used to have to make videotapes that I would hand to the media. So if there was an announcement about cancer or malaria, I would transfer video, like microscopy and animations, and make lots of cassettes and hand them physically to the journalists. cameras. So that's where it all sort of began. So like the monitor in the middle there was, anyway, I was very, very proud of the fact that I had that there. So I'm now going to play some animations from 20 years ago, 2002. How my career really opened up was Melbourne Museum was being built in the late 90s. And I think it launched in 1999 or 2000. And I was invited to contribute an image, a single image, a black and white image of DNA for their new exhibition of the body. And I was just delighted and thrilled to get a single image of DNA. I downloaded the PDB and I created an image and gave it to them. They put it up on the wall. And it turns out the curator there ended up in Boston, working for at Cold Spring Harbor Lab for James Watson, the co-discoverer of DNA, and so on. And they were going to create a 50th anniversary Discover the Double Helix TV series, and they needed someone to create DNA images. And so I was invited by them, because they knew of this image I was creating here, to come and be responsible for all the molecular graphics. And so it was a 5-part TV series, and I was very, very fortunate to work with an incredible team in London of documentary producers. And it was really them that won the Emmy, and we did lots of different outcomes, education product, which won a BAFTA. I mean, it was an incredible team, and I was very, very fortunate to be part of that. So this is the animations from that, or just a brief taste of that. And it's helix structure. And what we're going to do is look at its physical structure of DNA, which is made up of, if we unwind the helix, double helix, and then unzip it, we see that DNA is made up of these 2 strands. The things that look like teeth are the letters of the genetic code, but these 2 strands chemically run in opposite directions, which make a number of complications for your cells when the genetic code is being read, or more particularly when the DNA is being copied that you're about to see. So again, this is as accurate as we can make it in 2002, yes, for what the actual copying mechanisms inside the molecule are. So DNA is entering the production line from the left-hand side there, and hitting the collection of enzymes that are ripping apart the double helix and making an exact copy of each strand. One strand can be copied directly, it can be seen pulling off the right-hand side, but things aren't so simple for the other strand because it must be copied in the opposite direction, which is the version your cells do. Thrown out repeatedly in these loops and copied one section at a time. The end result is 2 new DNA molecules. Now this is being presented in real time as it would be occurring inside your cells, at least 2000. I mean, there's a whole bunch of simplification going on there to make it watchable, but broadly, this is the structures, the shape, how they're interacting, broadly accurate, at least for the science in 2002. In fact, it was this animation, so it became part of the TV series. It was actually, this animation was invited to many art galleries around the world. And it was this that Björk actually saw and contacted me out of the blue in 2010 because she wanted to create a DNA replication drum machine for her new album. And we did that. And you know, so, and to rewind again, when I was a teenager with my Amiga, so Björk is only 2 years older than me, so I had a crush on her when she was, and I was buying her records, and so an email from Björk, you know, at any age was, oh my God, I was over there so quick. Yeah, we made a DNA replication drum machine, a bunch of things, and also a stage show and a music video and all sorts of things. So it's a fantastic, so stick with science, kids. It could lead things to, you know, big surprises. Yeah, so anyway, this animation was really the one that opened up many, many opportunities for me. And one of the other things I'd like to also reference is it was the first time I'd ever encountered flexible proteins, which were those arms, and the literature sort of referred to them like I'd never seen anything like it before, but I built it because that was what's in the literature. And this will particularly resonate with the more recent animations that I'll show you later on. The animations I'm now going to play are from more recent production, which we published in 2021, last year, which was about a 4-year project where we created animations of the complete respiration sequence. So from glycolysis, citric acid cycle, electron transport chain, and ATP. We created a whole suite of animations, step by step, every single enzyme. I reconstructed what they looked like, what the chemistry would look like. And so it's a complete suite of animations which we provide for free for science education around the world. And I'll just be picking out a couple of those animations and showing you what they look like and the science that went into producing them. Just briefly, this is our pipeline, which we have developed over the last 4 or 5 years. The most important stage of all these animation production is always the research. That's where we put our time, majority of our time, and looking at all, we do a literature review and we chase up all the different sorts of data, the protein data bank, so X-ray, crystallography, 3D models, molecular dynamics simulations wherever we can, cryo-EM tomography, 3D reconstructions, all of that kind of data, which bring straight into our animation software, which in my case is called Maya. It's a big animation package that's used for Hollywood special effects, movies, video games, and so on. Once we bring in all the data, we integrate it together, all these different kinds of data, we can bring them together as a single visualization. We then export to a game engine, which in our case is Unity. So all of our animations now play live real time in game engine, which creates a number of opportunities. But most of my work is I'm called upon to create movies, linear movies, but now we create them in game engine. They're playing in real time on a game PC. We need animation. So I just create a virtual camera and just film what I'm seeing. But essentially, it's all real time. So we can go to augmented reality and virtual reality. It's all in game engine all the time. Just a little bit of the source of data that goes into building an animation. A longtime collaborator of mine is Dr. Mike Kuiper. He used to be here in Docklands at CSIRO Data 61. He's now up in Canberra, I think at ANU. He specializes in molecular dynamics simulations. And the molecule on your right is ATP, which is from this simulation here of ATP surrounded by water molecules. And what we're trying to do, so I take Mike's data on the left, I'm able to extract out ATP, I've colorized it and prepared it for animation. All the small molecules that you'll see in the animations have gone through this process. I'm trying to capture all the molecular vibrations and wiggles and the dynamics you don't normally see in this sort of molecular visualization. The topic I'm going to focus on is on the electron transport chain. This is my favorite figure of the electron transport chain from a paper in 2017. What you have is the 5 different complexes of the electron transport chain, complex 1 on the left through to complex 5 on the right. We're going to pull out 2 of these and we're going to look at visualization animations that I've created for all this. From this paper, what they're doing is showing you they're using the protein data bank models of these particular complexes, and the blue band through the middle there is supposed to represent the plasma membrane. So, sorry, the mitochondrial membrane. These are all embedded in the interior of the mitochondria. And so what they've done with this figure is they've overlaid with just the chemistry. So little arrows showing this reactant going to this product. And what I want to do is reconstruct not only what the chemistry looks like, but also the landscape. Where are these enzymes are located, and to reconstruct the membrane. So to give a sense of place and location for students. Next level of data, this is from a paper where they were looking at the E. coli bacteria ATP synthase. So this is complex 5. So okay, I'll go back. So we're going to focus on complex 5 on the right-hand side there, which is a very popular enzyme complex for students and also for animators to create because it's such an incredible rotary motor. So this is from our paper, and what they've been able to do is capture the ATP synthase in different conformational states and get the structure out. On the left-hand side, they've created a figure of all the different enzyme parts. And on the right-hand side is their movie where they're morphing between these different conformational states. They only had 3 conformational states. I actually think there's 6, but the 3 key ones they were able to capture. I take this data and I'm able to import it directly into animation. This is a beginning of building an animation. This is my early model of ATP synthase, where I've taken their data, I've added B factor and other things, but also the chemistry. So I've got little, the reactants coming in, landing at the enzyme and reacting and so on. So this is the next level, or the first level of building these sorts of animations. Wherever possible, I will always show things in, quote, real time. ATP synthesis, this rotary motor inside human cells, produces about 80 ATP per second. So I've made this emit 80 or create 80 ATP per second to give a visualization of that. But of course, at the nanosecond time scales, the ATP would emit it, and then a next frame would be a kilometer away at this scale. Like so I've had to slow it right all the travel of these little of the ATP to make sure they're all visible within the frame. So it's a compromise of time scales to convey some information like this. The next level of data, this is from a paper where they're looking at ATP synthase and showing that ATP synthase forms pairs in the membrane. So this is embedded in the mitochondrial membrane, and it creates a pinch, a kink in the membrane, which, as you can see on the left-hand side from this electron micrographs, all the measurements and the angles and such things go straight into building the animation. The next level of data is this beautiful electron microscopy, which shows that the pairs of ATP synthase form these rows of pairs. And what you have is different species. On the left-hand side is a cow, 3 different types of yeast in the middle, and then the potato on the right-hand side, showing all these little variations and differences between the species. I love those sorts of details, but particularly the electron micrographs up above, showing these rows of pairs running along these membranes. All the different measurements and so on go straight into building the animation. So this is my next level of animation, where I'm creating rows of ATP synthase. And this is starting to develop it also for classroom use. So the teacher can show different structures and present it however they want, with their own language appropriate for their audiences and the students. But we'll show you a different version in a moment. Next level of data is looking at cryo-EM tomography, where they're reconstructing the inner membranes inside a mitochondria and showing the location and the rows of ATP synthase, and all that sort of good stuff goes straight into building the animation. One last bit of digression, I guess, is I care very much about the art, the cinematography of my animations, but also it's actually the emotional feel, how you feel about what you're looking. And a big part of that is the use of color and sound design. Color evokes emotion, but also particularly sound for making something come alive and grossing. And a big part of what I'm trying to do is make kids lean in and lean into, oh my God, ugh, all that sort of good stuff makes it memorable and also makes them pay attention. So in this animation about to play, an artistic embellishment we applied was also music. The gentleman on the right-hand side there with the blue shirt is Frank Tites. He's a buddy of mine since university days. He's now a Grammy-winning artist, and he does film scores for feature films in Australia and around in the US. And this is the middle school choir from the school my kids go to. This was in 2018, I think it was, or 2019, before COVID hit. And we recorded a musical score, because I think this rotary mechanism is pretty wondrous, and I wanted people to have a sense of awe about how incredible this stuff is, much like a cathedral, or how you have a worship of just the splendor of architecture. That's what I wanted to evoke a feeling of for this animation. So yeah, this is the animation. It's very much aimed at high school kids. I want it to be narrated with the voice of someone of their age, and then it has all the rest of it. So I'll play and I'll talk about it afterwards.
All life on Earth depends on this tiny, energetic molecule, adenosine triphosphate, or ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA. Generating ATP for your living cells are mitochondria, electrochemical batteries that converts energy from the food you eat and oxygen from the air you breathe into ATP. So where does your ATP come from? Deep inside your mitochondria, rows of molecular motors generate ATP, the molecule essential to all life on Earth. Enzymes bring together reactants to form a chemical bond, converting mechanical energy into chemical energy. A ring of enzymes work in step, creating 3 molecules of ATP with each cycle. Inside the molecular motor, a rotating axle powers the sequence. The axle is attached to a rotary molecular motor, moved by the force of protons pushing from the other side of the membrane. A difference in proton concentration propels the molecular mechanism. Synthesis of ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA.
So that was just one version. We do many versions of these animations, different languages, but also ones without music, if that offends people. We're prepared for, different kinds of audiences. We provide with, or no narration, so the teacher can actually speak over and do their own presentation. So we do all of that. But all of that was done in Game Engine and plays live. This is the raw animation. So again, this is what it looks like on a normal gaming PC, everything is built on loops, seamless loops that are like the gaming technology. So everything, even the small molecules, the camera move, everything, this whole scene is a seamless loop and can play endlessly. So if it was in Game Engine live, the whole thing just plays continuously as long as you need. But I've also made it so this movie, if you have it on a seamless loop, the camera, everything else, it just will continuously play. That is a big change for me, because in the past we created animation. It used to be extremely difficult to create any kind of movie, takes a long time. But then that's it. That's the movie. Where we're now with this kind of thing, we can generate easily as much animation duration as you need for a video edit or for different productions. So all of this is automatic. The camera moves through and does a transection through the membrane, all of that. So it's a very, very different way, a huge advance for the way we produce our productions. Moving on. Okay, so this is hopefully play. So this we did in late 2018, early 2019, which is that same ATP synthase molecule, the first, all the animated ATP synthase is in Game Engine, so we can go export directly to augmented reality. I brought my iPad along if anyone wants to play with that, but essentially the idea is every kid has a phone in their pocket or a mobile device, and the idea is they can create an ATP synthase there on their desk and then look around it, and they can ask questions and they can do a group investigation of the ATP synthase. To me, this is mind-boggling that the technology, for the last bunch of years, all the iPads and phones are extremely capable. They're like, I thought, it's like 500,000 atoms. Is it going to be able to load the ATP synthase? It effortlessly does it. Not only that, you can actually keep on pressing and create 10, 20, 30 of them. They all play. It's like, we're only beginning to, exploit this kind of power and capability. So I'm very optimistic about that. This is going to be our future, is doing more interactive, real-time playback rather than just movies. Okay, I'm going to play one other animation from the electron transport chain, and the one we're focusing on is number 2, complex 2. It is called complex 2 of the electron transport chain. It's also part of the citric acid cycle. What if I got a laser? Nope. Don't know. I'm gonna press the wrong button. There we are. So this is it. succinate dehydrogenase. And what we're going to particularly focus on, I was very interested by this reaction here in the membrane, Q going to QH. That stands for quinone. It is a carrier molecule, a coenzyme in the membrane, and it carries electrons from one place to another. And I really wondered what that was all about. Like, how does that work? And that's really, it was certainly a revelation for me. I learned a lot from this animation. Is that me? No. So again, we begin with a plasma membrane simulation, molecular dynamic simulation by Dr. Mike Kuiper. The gray molecule, the ball and stick molecule in the middle there is quinone, coenzyme Q. I can't do anything about that. So I was informed and learned from how the coenzyme Q was behaving in the membrane by sitting across the double lipid bilayer. And it also informed me about where the electrons would be carried and passed on. So anyway, very informative to me. I took that data for the membrane as well as the coenzyme Q and applied that to the animation. This visualization hopefully will play. It's not going to play. Okay, so this is an animation, a molecular dynamic simulation. And the reason I had it in there is it's actually showing ATP and a kinesin motor, an enzyme. And when you do a molecular dynamic simulation to show how ATP comes and docks there, you have to wait a long time. And this movie ran for about 90 seconds or so. And each time step is a nanosecond or billionth of a second. And you have to wait around quite a long time for it to randomly wander around stochastically and arrive at the enzyme location. But what you do see is also the dynamics of ATP, the molecular vibrations, and you see the stochastic nature. I very much want to emulate the look and feel of it arriving stochastically, but I needed to move along for storytelling. As an animator, we need to tell a story in a reasonable amount of time. So it's part of the challenges of doing these kind of visualizations is trying to retain the true look and feel of a molecular dynamic simulation, but to actually tell a story. So that's one of the big challenges. So hopefully this will play. Okay, so this is, labels are over, it doesn't matter. So this is complex 2 electron transport chain. It's also step 6 of the citric acid cycle. And it's an enzyme that's embedded in the inner mitochondrial membrane. And again, this animation, everything's playing as a loop. It's actually a 10-second loop. The whole thing is a 10-second loop. It can play endlessly as long as you need for teachers or students to spend to study the various mechanisms. So the chemistry begins up here where succinate docks in this location here. It releases 2 electrons that then quantum tunnel all the way down to coenzyme Q, which sits here. And this is where, so coenzyme Q and the 2 electrons are received by coenzyme Q, which then travels in the membrane and goes to other enzymes, the electron transport chain. What else can I say? So what's very important to me was to create a location. These are all the other enzymes of the electron transport chain, but also to reconstruct all the small molecules, or at least a sense of what the small molecules would be like in this kind of visualization. So a 10-second loop plays endlessly. And moving forward again, this is the final one I'll play from respiration. This is a gigantic molecular enzyme complex called pyruvate dehydrogenase. It is the enzyme that sits just after glycolysis. It takes the product of glycolysis, pyruvate, and prepares it for the electron breaks it down for electron transport chain and for the citric acid cycle. So it resides inside the interior of the mitochondria, but it's a massive complex of around about 80 different enzymes, or 3 different types of enzyme, but 80 different in total for this complex. And so this was a massive challenge to build. And I'm going to show you a little bit of the science and how I went around about building this animation, and also particularly the flexible arms. So whenever I'm starting off on animation, and it's almost always the case, there will be a David Goodsell illustration. For those of you who don't know David Goodsell's work, certainly his style is iconic. These are our watercolor paintings that he does. They're all done by hand, but exquisitely accurate. He does the molecule of the month for the Protein Data Bank, has been for decades. And he was totally my inspiration as a cell biologist in the 90s. When I saw David Goodsell's illustrations, I went, Oh, is that what you're talking about for molecules? Because it was it revealed to me what the molecular world was like. So this is David Goodsell's illustration of the protein pyruvate dehydrogenase. You have different structures which are from X-ray crystallography, but what's particularly interesting is these regions here, these tethers here, they don't appear in the protein data bank, but we know they're there. David Goodsell has laid out a map, a roadmap of how these things are coming together. And this is very informative for me for building my own visualization of this system. Yeah, and it's always a relief and a delight when David Goodsell's been there before to a topic. Pyruvate dehydrogenase has been studied for decades. This is back in 1964 with an electron microscope. They can see at the limits of resolution, this is a big complex. And this is from a paper and where they've drawn a figure of what they think the different enzymes and how they're coming together, the 3D structure of what pyruvate dehydrogenase is like. Moving forward to 1979 with better microscopy, they are now starting to see sort of more of the shapes and how this complex comes together. And that's their updated illustration of what they think the pyruvate dehydrogenase is like. And moving forward, this is from cryo-EM tomography, 3D reconstructions from slices of EM, where they're reconstructing the 3D with incredible detail, the interior and the exterior from cryo-EM of the pyruvate dehydrogenase complex. So all of these, the locations of everything, this is absolutely gold for building my complex. And actually, this was really the foundation of my own model of the complex. So this is the animation. It's actually just after glycolysis. You've gone through 10 different steps of enzyme. And at the finish of the glycolysis animation ends up here. You're still on the exterior of mitochondria. And I'll play the animation and just talk about it afterwards.
The product of glycolysis, the pyruvate molecule, is the source of carbon and electrons for aerobic respiration reactions inside your mitochondria. Pyruvate enters pores on the mitochondria membrane and is transported into the interior. The reactions linking pyruvate with aerobic respiration are performed by a huge protein complex made with multiple copies of 3 types of enzyme. The first enzyme catalyzes decarboxylation, transforming 3-carbon pyruvate into a 2-carbon acetyl group, generating carbon dioxide as waste. The acetyl group is then grabbed by the second enzyme, using flexible arms to efficiently transfer the product between reaction sites. In the active site of the second enzyme, the acetyl group is attached to coenzyme A, generating acetyl-CoA, fuel for the citric acid cycle. Two electrons from pyruvate oxidation are retained by the second enzyme before passing them to coenzyme NADH, catalyzed by the third enzyme. Coenzyme NADH travels through the matrix, delivering electrons for the electron transport chain. In a sequence of reaction steps, the pyruvate dehydrogenase enzyme complex generates fuel for aerobic respiration reactions of the citric acid cycle and the electron transport chain.
So that animation was particularly difficult to create, to reconstruct the flexible arms and the whole structure of this thing. And I did that must have been 2020, I think. I can't remember, 2020. And it took me a lot of time to crawl through the literature and build that. And it was less than 6 months after I created that particular animation that the AlphaFold came out. And what I'm comparing here is, play. My flexible arms. So this is enzyme 2, and it has these flexible arms and so on. And I reconstructed this by hand, adding amino acids, and it was just a pain in the ass and took me weeks and weeks and weeks. And then AlphaFold came out, and I ran the same enzyme on it. And now, of course, they're all available. This is before they released all of the structures. And oh my goodness, there were so many details here that I'd missed, and the tethers were much longer than I expected. I do now understand also, though, that AlphaFold does exaggerate how extended the unstructured regions are. But if I had had this, it would have saved me weeks and weeks of time. And this is certainly the future. It's just absolutely golden for doing our kinds of productions. So really exciting times, and wild frontiers ready to be, marched into. Right, so that's the formal scientific animations. After this talk, I'm going to go about a block down that way to Science Gallery, Melbourne, which is part of Melbourne University, just on the corner of Grattan Street. And there I have an art exhibition currently playing. It's only running for the next 2 weeks. It's been running for the last couple months, and it's about to finish. And what you have here is virus 1 million times. These glass bricks embedded in the building facade are able to magnify the world by 1 million times. And if you look through each glass brick, you'll see one of the 10 deadliest or most infamous human viruses, HIV, herpes, papillomas there, monkeypox is there, COVID, of course. So as you look through each of these glass bricks, it's their magnifying device able to magnify the world by 1 million times. And so for each of these viruses, if some of them have their genomes spilling out, so RNA or DNA, they also have antibodies coming in and clearing them out. So if you were that is of interest, seeing science being taken into the art area, this is running just for the next 2 more weeks, and I'll wander up there, and if anyone's interested after this talk, and happy to answer any questions there. So I'm very, very fortunate to have been invited to participate in this project and many other projects here in Melbourne, around the world, of bringing science into the public sphere and recontextualizing it to make it accessible and engaging for the public. Another artwork is also that I installed 15 years ago at WEHI. Is called the Illuminarium. So this is just on the other side of Melbourne University, less than 500 meters away from here, is the Illuminarium. And what it is a 6-story tall artwork that plays microscopy, real microscopy from the labs at WEHI, and it plays a non-stop roster there. And the idea of the Illuminarium is to act as a beacon to draw people's eye, to look over there and to admire the beauty of this kind of the research, the real data, and to make people wonder what's going on over the Institute. So yeah, that's another one of my artworks I've done, and which I'm really delighted, and always it's very exciting and interesting to try and bring science to the public sphere. This is my team. I can sort of see, poor Tsuko's covered over. So Justin Muir in the middle there has been with me for 4 or 5 years now. He comes from 20 years in the games industry. He's always also a biomedical animator for a couple of years, but Justin Muir worked on Fear, a big blockbuster, and also Mortal Kombat. And he has completely transformed our pipeline, and all our game technology stuff is, our entire pipeline is due to him. And it's really tremendous and very, very exciting where we're heading now. And Etsuko, who you can't see quite there, Etsuko has been with me for about 14 or 15 years, I think. She's also got a strong science background and specializes on doing animations on the topics within WEHI. If you want to see more of our animations, our YouTube channel, all our animations are downloadable for free. There's our details there. But with that, I will wrap up, and thank you for your attention. And I'm happy to answer any questions. So thank you.
I lost more high ground as far as screen time is concerned. Are there any questions? Yes.
I'm a big fan. Thank you for the presentation. I'm wondering, first of all, why do you continue to use my name instead of, for example, using Blender and other
Yep.
Considering the newest updates.
Yep. So Blender is amazing, and that's one I recommend for any students or anyone getting into it. I use Maya because I've been using it since 2000, and our pipeline works. And if I was going to switch, I would switch to Side Effects Houdini. It's the one I've a long time admired. It's the one. It's like, oh my goodness. So Blender I think is great, and I literally just had an invoice for almost $10,000 worth of Maya license, or every annual license. And I go, why are we doing this? And it's like, well, our pipeline works, so I'm not going to switch. But Blender's excellent. It's not Maya level, but it does. I see most of the stuff I would look for. Yeah. So, and then Unity again, that's one we chose a few years ago. It's all excellent now. I mean, we could switch, but it would just take time.
Thank you. Fantastic talk. I was just wondering, because visualization is such an integral part of communicating science, not only to the general audience, but also like in our papers and journals and so on. Do you believe that research labs or institutes should be hiring dedicated artists or designers, or should academics themselves be investing into developing these students?
It is a challenge because it is a whole different skill set to do visualizations. There's a lot to it, and animation is a whole nother level again. But fortunately, like I showed David Goodsell before at the Scripps Institute, they have developed, so if you want to do molecular illustrations particularly, they have developed an app that's free that makes you able to draw and scribble, like you create your structure, you import your it does it automatically. It's fantastic. So you can create David Goodsell illustrations easily without any skills. So I think that's a huge advance. There are a lot of different programs like that trying to put tools to enable anyone to be able to create these sorts of visualizations. And Cell Page, I think it's called. It also does animation as well, like Brownian style. So perfect for illustrations for figures for journals, but also public outreach. There are growing numbers of places for science artists here at Peter Mac and Garvin and other places, but then particularly in the US that's where most people are. Most people who do what I do end up in advertising, doing toothpaste ads. Or shampoo ads, and so that's that's where the money is. It does require an investment because it does take a lot of time and expertise to do this, but I think there's an strong appetite for it from different institutes and so on. I tend to tap into global or large philanthropies that are wanting to promote science and get good science education tools in the classroom. That's where most of and science centers and museums and documentaries. But that's a pretty limited area, like for funding, but that's where I've existed. Yeah. Yes.
Kind of working on me, you're an inspiration to everyone else. Well,
oh, thank you.
Thank you for all this work. However many years.
Yeah. Thank you.
I work with the Unity engine a lot in my spare time, and what is the most interesting intersection between those two fields for you, and how might you recommend someone like myself to blend them more?
Oof. There's the technology in the pipeline and the techniques. So I would look at Blender. In fact, you're already in Unity. I don't understand what Justin has done. Like I'm really into the technology, and I don't know. He talks at me, and I go, but I think someone like with your skills is really, it's like huge. It's happening, like this potential is huge, and there's so much need. So, but also to tell your own stories that you may want to explain. There isn't any I don't know of Unity style tutorials that work for this kind of stuff. Like I was for Blender, and there's Brady Johnson, who's over in Perth, who is he's going to end up in Europe real quick, I expect. But he's doing tutorials that explain how to bring this into Blender and how to bring in your data or your molecules and how to do everything that I'm doing, quite frankly. So if you want to learn that's good. I don't know. It's sort of you have to be like you are into it and just do it on the side. I was very much a hobby on the side, and then just opportunities came up and I just did it because I wanted to, and I'm passionate about it, and it gradually grew into something. So yeah, I don't know. There's no formal way, as far as I know. I don't know. Yes.
So once again, sorry, what was your colleague's name?
Brady Johnson, the Blender guy. He does YouTube tutorials. He's what everyone talks about. So he's Blender, and he has excellent tutorials and tools that he's built to bring in and do molecular animations and molecular dynamics looking stuff. So Brady Johnson.
Recent technology in using the field of AI open the doors to generating images using text or through data diffusion. I'm wondering if that's going to affect your job in creating these very unique animations, and so it would be using that technology.
I don't. It's moving so quickly. You go, oh, sure, no way. But the way it's going, it's really quite staggering. I do think, though, that stable diffusion and those things are just creating a look and feel, emulating what these things look like. Where what we're doing as humans is I'm having to interpret the written papers and understand the data and build from, like, it's not a, so I think I worry for artists in general that they've been sampled, and this stable diffusion or those sorts of systems can create imagery that it's fantastic, and it's instant and free, where I do think there is a place for specialist artists who really understand a subject matter and are able to surf it, navigate it, and take in and reinterpret it. So that's the diminishing place for artists right now. So but I who knows what's coming next, because it's pretty staggering already. Like, yes.
Yeah, maybe just following on from that, you mentioned at the start that you always use the data in the literature as your sort of baseline.
Yep.
Things like, I suppose, alcohol coming on the scene.
Yep.
Maybe that would be more of a baseline that we put in the simulation. So I'm wondering
Yep.
if you see scope like that the other one. Maybe some of these simulations started to actually more than violate.
It's true of model building, I guess. And if you're building from, like, I don't think my work does not present a model that almost never, which I haven't actually already read in the literature as an informed speculation and discussion in a good review paper. They'll tell they'll fill in the gaps. But when I build it in 3D, there's often a case where it can't reach, or this enzyme can't, like they're describing it, but they haven't put it together yet. So I think there's value in that. But that's true of a sketch, like having a napkin and a pencil and sketching it out. It always changes the way you think about it. So I wouldn't want to overemphasize this being a tool for discovery, but I do think it's important for representation of what we're thinking. So I don't know. It's in hands of an expert who understands the science and trying to. I think they could use it for model testing if they wanted to. But all of my stuff is emulation. It's not real simulation. It's look and feel, and it's telling a story. Is the purpose.
We're talking about sketching. Does low-fi or low-tech artistic ability support your work, or can you get by without being able to draw or paint? How? Yeah.
So I can't draw at all. I need to have an undo button. I have to have a mouse and undo button. It depends on your skills, and a lot of these packages are very geared towards programmer types. I'm not a programmer either. I don't program. They're multi. It depends on your talents and what you bring to bear, and I guess. And I've been fortunate. Like Maya is a fantastic package for however you want to work. It'll provide the tools. So that's where I've been.
Yes. Yeah. I can only assume 20 years ago that there was a lot of manual labor when there was a lot of these animations. Do you feel that with your animations that you've produced in the past few years that there's been more automation to it, or are you still having to go in and tweak individual methods?
Yes, definitely. We've moved into, yes, you're right. Early on, it was all manual. And definitely in the last 10, 15 years, it's moved on to, it's fully generative now. I generate And all the soup of molecules and the way the enzymes come in is all spontaneous, and I can change a seed value and get a completely different scene from the way we build it. It gets out of my hands. And so, yeah, I think that's a big deal. What else has changed? Yeah. Anyway, so it's the technology or the tool set or your paintbrush determines what you're going to create. So yeah, but it's all generative, and with procedural. Yes.
Do you have any tools that you wish you could try and streamline your process in animation?
I hope not, because I really enjoy the process. Like, I enjoy the struggle and the research and the just wallowing in the science and going for a walk and going, wow, you know, like, oh, I can't believe it. So that's a big part of it, is just the internal thinking it through. The tool sets are it's getting so good. So actually, to go back to the previous question, also back then it was manual, but also rendering, creating the image by the computer was a big deal. And literally in the 90s, you'd create a molecule, like the DNA molecule I created for the museum, and I go click go, and you'd watch the lines go across the screen. It would draw, and I had to wait until it hits the molecule. Like, oh, no, it's wrong, stop. And I'd start again. And you just watch the rastering of the lines. For the DNA TV series in 2002, I had a rack of PCs. We had 100 grand worth of PCs to render the images. And I would give the machines a job to render. And one of them in particular took 10 days. So you go, you wait 10 days, you press play at the end, go, oh, it's the wrong color. Or, oh, that was the one I meant. And you have to wait another 10 days. Like, that would keep a machine busy for 10 days. And I was repeatedly. So rendering used to be the complaint of animators. We'd be sitting around waiting for the images to be produced and then rendering and re-rendering. Now it's in real time. Like, that's gone. So, you know, like, manual labor, it's all generative, but it's also, it looks good. Like, you know, just record that. It's not as good as slower techniques, but it's good enough for me. Like, I'd much rather the speed and time rather than going for the fancy super high-end graphics, which you can get from, but it's all catching up now. All the fancy graphics are now in GPU, and it all looks good. So yeah, no, I've been very, very fortunate from the 90s or late 80s all the way through the revolution and gaming technology, all these forms of technology. It just hasn't stopped. And now I don't even care. Like, it's all possible. Like, I used to be the new thing would come out, new Mac would come. Whoa, it's so fast. And anyway, so yeah, it's all, I enjoy it. And it's been surfing that long run of technology developments. And which is impacting science as well, with AlphaFold and all that stuff. So it's all the same. Yeah.
Just one last question from that corner.
Your pathway is talk about how the manual aspects has started changing from more generative style. Have you felt that has negatively impacted your artistic intent with relationships?
Not at all. I'm very look forward to the spontaneous surprise I get when I run it, and I go, oh yeah, you know, like I'm glad I like it out of my hands. And there is a lot of control you can have for the aesthetic and the cinematography. And then when you get into Game Engine and Unity, you can then control lighting and do all sorts of choices live. So it's just a matter of, tweaking. And so it's just it's such a profound difference to what it was, of course, 20 years ago or 30 years ago, like, and the future. And so all the young kids coming up this day they're native to devices and screens and creating movies and editing on their phones. And so when they come through, like, I'm a dinosaur, when they come through with their skills, your skills and your knowledge of a special area, I'm very optimistic, but it's more, again, it comes back to the funding and who's going to pay for it, the time. But I think more and more opportunities will come along. So, and as a recognition or desire for these kinds of materials, particularly for public outreach. But yeah, there's no particular formal way of doing that in Australia, for example. That's why I keep going to the US. There's definitely lots more opportunities for money to put teams together and make this happen. So, yeah.
I think this was a wonderful talk.
it just showed how combining science and art allows us to draw in another generation of scientists and artists, hopefully as well, and how the combination allows us to understand stuff that without, in isolation, would not be able to understand, comprehend, or enjoy. So thank you very much for this artificial experience and for this enlightening talk and for the time that you donated to us today. And yeah, thank you very much for all the patience.
Thank you. Thank you very much. Thank you, Michael. Thanks. Thank you.